Hi, this is Dale with Behind the Bench. Here we are at Genomic Solutions Tour, San Francisco, and this morning we heard from Brian O'Connor from National Jewish Health. Brian, really enjoyed your talk today about epigenetics.
Thanks, Dale.
What can you tell us and our audience about what you just talked about?
Yeah. So we're really excited because there's this constant debate of nature and nurture. I think we've gotten really good as a scientific community looking at genomics and genetic contributions to disease. But there's the other side, the environmental side, that we really don't have a good handle on. So in this nature and nurture debate, again, done a really good job with the nature part, but not a really good job with the nurture part. Epigenetics, I think, is a really exciting field. It's kind of a young science. But it really creates that bridge between environmental exposures and molecular biology and what could potentially happening inside of our bodies.
And specifically, you're looking at asthma.
At National Jewish Health is a respiratory hospital, so we focus on respiratory diseases, and asthma is sort of a poster child for one of these environmentally regulated diseases. We know in children asthma rates are rising. We don't really know why, but we have a strong suspicion that it's the environment. So we'd love to understand what those environmental exposures are doing to children at a molecular level.
And you showed some data regarding your early efforts at ChIP-Seq on the ion proton.
Yeah.
What can you share about that?
Yeah, so this is tremendously exciting. So ChIP sequencing is a process where we can create what we can call an epigenetic signature in specific cell types. And these signatures really point to places in the genome where there's an environmental event, something that's happened to the genome from an environmental exposure. And so using the ion proton technology, we've been able to, and working with Life Technologies, we've been able to optimize this process called ChIP-Sequencing. We can now use it with really small amounts of cells, which is very important for working with human samples, because oftentimes you can't get a lot of cells. It's not like working with a cell line where you can grow up tens of millions of cells. Here, whatever sample you get that's it. That's what you have to work for. So with ion proton technology, we can do ChIP sequencing and create some of these epigenetic signatures with these human samples.
If I remember your data, you had 20 million cells all the way down to 1.
Yeah.
And you're able to show some reproducibility, right, from these flow-sorted samples.
Extremely good reproducibility. When we first started this process, this is about 3 years ago, there was no reproducibility whatsoever between 20 million and 1 million cells. 1 million cells is sort of our holy grail because at that level we can really begin to do some of these human samples. So now, where we are today, working with Life Technologies, we can get very high reproducibility between 1 million cells and 20 million cells doing ion proton ChIP-Seq.
Yeah, and there was a question from the audience afterwards about some of these publicized low input ChIP, and you had some input about that.
Yeah. So there are quite a number of publications out there and other companies that sell kits saying you can go down to 10,000 cells. Some of those things are just not real world. In the real world possibilities, a lot of times when they say 10,000 cells, what they actually mean is 10,000 cell equivalents of DNA that they've already isolated. Sure, once you've isolated the DNA after the ChIP process, you can titrate it down and show that you can get some libraries. What we're talking about here really is, what if you only have 1 million cells to start with or less? Then DNA equivalents don't mean anything. So we're really excited about this new protocol because it works for those low input cells.
I look forward to seeing that. You mentioned it's going to be written up in an app note pretty soon.
Application note. Absolutely. So we've worked with the folks here in South San Francisco. They are putting together an app note. I've seen the proof, so it's just about ready. It should be on the website very soon. It has all of the details about some of the tweaks that we've done and really details the protocol.
Yeah, one of the last things I wanted to ask you about was, as far as these samples go, you showed some cell sorting in terms of phenotypic markers.
Absolutely.
Can you comment on that? Because you mentioned GWAS and the genomic side.
Yes.
But here you're looking at protein markers.
Absolutely.
And separating cells on the basis of that.
Absolutely. So one of the things we didn't get a whole lot into, but is very important for us, is oftentimes it's not just about your ability to isolate a sample from a human being, but it's that you really want to look at a specific cell type, whether it's a specific immune cell type or a specific epithelial cell or a cancer cell versus, say, a non-cancerous cell in that subject. And so one of the ways that people do this today is by flow cytometry sorting. That itself presents some problems for ChIP protocols because oftentimes the types of assays you need to do and preparation you need to do for flow isn't always compatible with ChIP analysis. So we do describe as well in this app note that we can fix our samples in the way you would do for ChIP. That's your first step, actually. And then we do our flow cytometry staining. It doesn't work for every single marker. That's something you would have to optimize. But for us, it worked for quite a few of those cell surface proteins. So now we have the ability to be able to link the phenotype of the cells, the activity of those cells, depending on whatever protein marker you're looking at, and then link that up with the epigenetic marks that you come up with later on through ChIP sequencing. So it's a very powerful assay when you start thinking about the implications of histone modifications representing that environmental change, and you've already got the inherent phenotypic data from your flow sorting built into that. So when you put those together, in terms of sort of clinical research, understanding disease at a new level, this is unprecedented.
That's great.
Yeah.
Certainly your enthusiasm and excitement came across in the talk.
Yeah.
Thank you very much this morning.
Yeah.
Well, to continue to follow our activities at the Genetic Solutions Tour, follow us at lifetechnologies.com/behind the bench. And if you have any questions for Brian, please feel free to leave them in the comments.